Hi everyone, this video is a solution video to the in-class assignment we did on blood pressure and studying blood vessels. So what you're seeing on the screen, this is the worksheet that we used. I split it up into different groups to tackle these things. We didn't get through everything, but the goal of this was really just to step through kind of a logic-based thinking, deductive thinking to kind of solve some problems. One of the things you see here is when you answer these questions, it says use a certain slide, right? Well, you can find those slides when you go to the page where you found the worksheet and click on worksheet activity images. And when you click on that, this is what you're going to find. You're going to find a PowerPoint analyzing mean arterial pressure. Okay, and so these slides were curated to go along with this worksheet. Okay, so question number 1. What two organ systems control organs extrinsically? Again, how are we controlling an organ system with an outside organ system, a different organ system? And if you remember from earlier lessons, we've been talking about this for most of the semester. What controls organ systems extrinsically is the nervous system or the endocrine system, and oftentimes both. Okay, again, it's a control from the outside of the organ system. Okay, your heart is controlled by sympathetic nerves, it speeds it up. It also is controlled by the vagus nerve, which is a parasympathetic nerve that slows it down. That's extrinsically controlling the heart. Epinephrine is a hormone that can speed up the heart, extrinsic. Okay. Otherwise, the heart has its own rhythmic system for action potentials, right? Those are from the autorhythmic cells, and that's intrinsic control. That control is coming from within the heart itself, its own properties. Okay, so that was just kind of a baseline. You didn't even need the chart. What kind of effect does sympathetic activity have on arterioles? Okay. Well, if you look at the chart, again, we have to go to number 3. There it is. Okay. Look down here. It says sympathetic activity exerts generalized vasoconstrictor effect. So that's something to remember when the sympathetic nervous system is acting on arterioles. What they're doing is they're causing them to vasoconstrict, right? That's going to reduce blood flow, but it's going to increase pressure. Okay, it reduces blood flow to the organ, but remember, we monitor pressure within our blood vessels, so it increases the pressure within our blood vessels. Okay. All right, what hormones affect arterioles? And that's, you can see here along the side. So these hormones we haven't gone through specifically. You don't need to know their specific actions yet, other than it's an extrinsic control. Okay. And notice these hormones all produce a vasoconstrictor effect too. Okay, so vasopressin is a hormone. We're going to actually discuss this a lot when we do the kidney. Okay, but it's a really widespread hormone. It's got a lot of different functions actually. So in this case, it has a vasoconstrictor effect. Angiotensin 2, another hormone. We're going to look at this also when we do the kidney. Okay, has a vasoconstrictor effect. And then epinephrine and norepinephrine. So remember, these are neurohormones that come from the adrenal gland. And we'll even talk about those more specifically. So for now, just list those hormones. Okay, next question. Name 5 ways arterioles are intrinsically controlled. Okay, and that's what you can see on this side. These light blue, it says local intrinsic control. So these are local changes acting on arteriolar smooth muscle in the vicinity. Okay, so again, local. Basically, if you put heat, right, in a certain area, that will cause vasodilation, right? It's a local control. The nerves aren't involved, hormones aren't involved. This just automatically causes, it acts on the smooth muscle. Okay, cold causes vasoconstriction. You reduce the blood flow. So that's one thing to list. Responses to shear stress. Now I asked the class, what is shear stress? Okay, shear stress means when, if my hand is the wall of an artery and blood is flowing through that artery, through the lumen, well, some of the blood will hit the walls of the artery, and that scraping across the artery is shear stress. Okay, so that is a signal to the local area that there's a change in blood flow. Okay, so it compensates for changes in longitudinal force of blood. It says right there. Okay. Next one, myogenic response to stretch. Okay, when blood flow increases and it pushes, there's more blood coming in, it's going to push on the walls of the arteries. And remember, arteries have smooth muscle around them and they stretch. Okay, so when that stress occurs, you don't want to get too much blood flow going to your organs because some of the capillary beds are so delicate you could just break them. Like blood flow to your eye. If you suddenly had a surge of blood flow going to your eyes, those capillaries could break. Okay, so there's a surge of blood flow, the arterial stretches, it constricts. And so it limits that blood flow from coming in. It constricts it right away. Okay, important in autoregulation. And we're going to look at that a little more closely in a minute. Okay, and then histamine release. There's histamine is a kind of a paracrine that's embedded in the tissues. And so we'll see some other local changes that occur when we go through different organ systems. Histamine can get released, right? And that'll increase blood flow. Really important in allergic responses. But again, it's embedded within the tissues, so it's an intrinsic control. Right now, I'm looking for you to understand the difference between intrinsic and extrinsic. Okay? All right. And then here it says local metabolic changes in oxygen and other metabolites. Okay, so what local metabolic changes is when the tissues, the cells of the tissues of that organ are becoming more active or less active, right? And so if you're more active, you're using more oxygen. Okay, and so we're going to look at that pretty closely too. But that's an intrinsic control because it's the organ itself that is changing its activity in order to, it's either increased its metabolism or decreased it. Okay, again, metabolism is just the sum total of chemical reactions that are occurring. Okay, and that's cell work. That's what cells are doing when they work. It's chemical reactions. All right, we're going to come back to these two in just a moment. Whoops, these two in just a moment. I realize I'm pointing at the wrong screen. Okay, so let's go back to the questions. So we finished with that group. So the next group, intrinsic control of arterioles. We're just going to look at more in depth at the intrinsic control. And we're going to use slide number 4. Okay. So now we're going to go through in particular these concepts of active hyperemia, okay, reactive hyperemia, alrighty, myogenic autoregulation. Okay, these are really important concepts for you to understand. Yes, study them for the test. Okay, and I think this flowchart makes it easy for you to understand and step through. Okay, hyperemia, all of these refer to A and B, active hyperemia and reactive hyperemia, have to do with substances as a product of metabolism. Okay, so let's go through active hyperemia first. So the question here is, explain the triggering cause of active hyperemia. What's kicking active hyperemia into play? What's causing it? What's triggering it? Right? And so the triggering cause is actually a decrease in oxygen and an increase in CO2. Okay, that's the triggering cause. But I want you to explain it. Okay, so why is there a decrease in oxygen and an increase in CO2? Well, it's because in that organ, that local area, those cells of that organ, they've increased their metabolic activity. They're working harder. Okay, and when you work harder, right, you have to generate ATP. So this is why I needed you to understand cellular respiration. When you generate ATP, remember oxygen is used at the end of the electron transport chain. You need to remember that. I asked the class that, and I got a lot of silence. So where is oxygen being used when we inhale it? Why do we inhale it? It ends up at the end of the electron transport chain in cellular respiration in the mitochondria, right? So it can pull those electrons across so we can establish a hydrogen gradient, chemiosmosis, to power our ATP synthase, and we make ATP. So we're using oxygen. Greater use of oxygen means you get a decrease of oxygen in those local tissues, okay? The other part of cellular respiration, when we're making a lot of ATP, remember we give off a waste product, carbon dioxide. So when we give off that waste product and we exhale carbon dioxide, we're producing a lot of carbon dioxide. Before we exhale it, that builds up in our tissues. Okay? So greater CO2 production means an increase in CO2. Okay? All right, so that caused our triggering cause here. Low CO2, low oxygen, high CO2. Okay, so we have receptors that can sense the levels of those gases, right? So that causes the local, so that decrease in oxygen, increase in CO2 causes the local release of vasodilating paracrines. Now you got to remember, what is a paracrine? Paracrine is a signaling molecule, okay, a molecule that's going to communicate with the cell to change its activity. Okay, in this case, that molecule is nitric oxide. Okay, it's a local vasodilator, so it gets released. That causes the smooth muscles. Okay, when nitric oxide comes to the smooth muscles of the arterioles, it causes them to relax. When smooth muscles relax around an arteriole, whoop, we get vasodilation. Vasodilation. That's the explanation here. Okay, explain how this produces arterial vasodilation. Okay, then explain how the result of vasodilation compensates for the original triggering cause and brings the area back to homeostasis. Okay, so we get vasodilation. That, step it through your mind. Use the flow chart if you get stuck. But if I vasodilate, I increase blood flow. If I increase blood flow, what does it carry? Oxygen. So it's bringing oxygen. There you go, increase oxygen delivery. It's also when that carbon dioxide is getting released out of the tissues, it's going to go into the blood. So that increased blood flow is going to pull it away faster. So it allows it to diffuse out of the tissues faster. So increased CO2 removal, which means it lowers the CO2. And there you go, we relieve this situation. Wow, that looked like a negative feedback of a homeostatically controlled situation. Okay? All right? That's what active hyperemia is. It has to do with just an increase in the local metabolic activity. Compared to the next question, reactive hyperemia. Reactive hyperemia, you can see starting here at the triggering cause, is pretty much the same, but the reason you have this triggering cause is different. It's not because of an increase in metabolism. Okay, cells are always active, at least even when they're at rest, they're still doing things. So you just have a lower metabolism. Okay, but in this case, there's an occlusion of the local blood supply. So let's go back to the questions and step it through. Explain the triggering cause of reactive hyperemia. Okay, so the triggering cause is the same. It's low oxygen and high CO2. But I want you to explain that. Why is this happening? It's because there's a blockage in the local blood supply. There's a blockage in the arteriole. We do that all the time, even very healthy people. We have plaque or a little fat that is on the inside of our arteries, and sometimes it falls. It can be deadly if it falls and blocks a vital coronary artery or something for any amount of time. Okay, but often it can dislodge, but we're going to see here how we get rid of it pretty quickly. So there's a local control that does that. Okay, so again, you get an occlusion, means a blockage. It blocks that blood vessel. It blocks that blood vessel. If the blood vessel is blocked, that means it's not bringing in oxygen. And again, the cell is still active, doing its thing, so it's using oxygen. But if blood isn't able to come in and replenish that with new oxygen in there, then you're going to deplete the oxygen. Okay, and in this case, the cells are still active, and now it's building up CO2. It's releasing that CO2 to the tissues, and there's no blood flow to sweep it away. Okay, so that's the reason why you have low oxygen and high CO2. Okay, explain how this causes arterial vasodilation. Same thing. Now these are the same. That decrease in oxygen, increase in CO2 is a signal that causes the release of vasodilating paracrines. It's nitric oxide. Okay, look it up. Nitric oxide. It says so on the PowerPoint slides in the lecture video. Nitric oxide produces local arterial vasodilation, and that can be in the arterioles that have the blockage, the occlusion. So when they vasodilate, boom, they open. There's more space, and that more blood flow is coming in, and it pushes that blockage through. The occlusion gets removed. That increases blood flow. You increase your oxygen delivery, your CO2 removal, and it relieves the situation. Okay. Next one. Okay, so that's how you explained it, brought it back to homeostasis. Next one says both active and reactive hyperemia use local paracrines to trigger the local arterial vasodilation. So what is a paracrine? Please look it up. It is a local signaling molecule. So one cell can release a paracrine that signals another cell. Okay, cells are releasing oxygen or releasing CO2 that signals the cells nearby to release nitric oxide that vasodilates. Nitric oxide is also a paracrine. What specific chemical acts as a paracrine in hyperemia? And that was nitric oxide. Okay, last one. Explain the triggering cause of myogenic autoregulation blood flow. That's this last one. Okay, myo means muscle, and it's an autoregulation. So the muscle, the smooth muscle, are going to relax or constrict depending on what we need here. So let's see what happens. Here your blood pressure falls, mean arterial pressure falls. Okay, so what does that mean? Step it through even before you look at the flow chart, but you can use the flow chart to help. Mean arterial pressure goes down, that means the blood flow to the tissues decreases. Right? Well, if the blood flow to the tissues decreases, there's less blood pressing on the walls of the arteries, so they're not being stretched. Okay, so decrease in the stretch of the arterial smooth muscle. Regular blood pressure stretches them a little bit, causing vasodilation. Okay, we saw that before. Stretch causes vasoconstriction, I'm sorry. That's how we just control our blood vessels. We have what's called tone. When the stress is gone, stretch is gone because the pressure was relieved. We have low pressure. Whoop, they're not getting the signal to constrict, so they dilate. Okay? Local arterial vasodilation, that increases the blood flow to the tissue, right? Which then relieves this situation of low blood flow. Okay, but the thing to remember is that this is a very temporary solution to our problem here. If mean arterial pressure is low for too long, then this is going to exacerbate the problem. It's not going to allow our mean arterial pressure to come back up. What it does is the problem is with low pressure, this organ now is not getting enough blood. So the immediate solution is to increase the blood flow. If this isn't fixing the upper solution here, it isn't fixing this problem, low mean arterial pressure. We have other mechanisms that hopefully are fixing that problem. Okay, maybe you need medication. Right, blood flow to the tissue remains normal despite the decrease in driving pressure. So again, it's a temporary solution. Okay. I'm going to end the video here, and I'll make a new video for venous return. Thanks, everybody.